Okay, so I'm here to present the automated recommendation tool, which is a machine learning algorithm that is ideally suited for synthetic biology. And if you go to the next slide. So ART is a machine learning tool that provides a predictive model. It takes your data, and the data could be, almost anything. ART is a very flexible tool that can be applied to a variety of problems. It could be things like protein concentrations, or a promoter combination, or media component concentrations, or maybe even protein mutations from what we saw in the previous presentation. Well, and then a response, and the response will be what you're trying to predict or optimize. It could be your production of biofuel or productivity of your bioproduct or your titer or whatever it is. Then it takes all that data, and if you can go next, you can touch, yeah, there we go. So it takes all that data and makes a predictive model that is probabilistic. So it tells you that this protein profile has a 10% chance of producing 10 milliamps per liter, or that protein profile has a 15% chance of producing 2 milliamps per liter, and so on. With that data, next, it makes recommendations. So using the predictive capabilities of the model, it tells you, well, decrease this protein number 1 to increase production, and increase the concentration of protein number 2, for example. And so that's what it does in general terms. It's very general, and we use for a variety of different projects, and I'll show you some of them. If you go next to the next slide, you'll see that ART is ideally suited for synthetic biology. Firstly, because it can work with very few instances. We've actually gotten a good results in active learning with as low as 27 instances, which is rather unusual. Next. ART also provides uncertainty quantification. When it makes a prediction, it gives you a probabilistic, a whole probability distribution of the prediction. If your probability distribution is very narrow, you can trust that prediction quite well. If your probability distribution is quite wide, it tells you that it doesn't really trust that prediction that much. And this is critical when you're dealing with a small amount of samples. And next. Also, ART provides recommendations, not just predictions, so it really gaps the learned to design phases naturally. Instead of just having a few predictions, it tells you, well, change this protein by this one, or increase that protein, or change that concentration, for example. And if you want more details, we have a webpage called art.lbl.gov. Let me give you one example right now in the next slide, in which we are improving titer of flavulin by optimizing media. In this case, the input is the media component concentrations. We have media with 15 components, 12 of which we're going to change, and we're going to see what impact that has on the flavulin titer. And again, ART will make a predictive model, and they use a predictive model to make recommendations to change the concentrations of each of the 12 media components in the next cycle. Next. In order to feed enough data for ART, we created a semi-automated pipeline that allowed us to create the new media in a high throughput manner. If you look at the top, we synthesized the media with a biomech liquid handler, and then we added the strain and the antibiotic and the inducer, and we fermented it in a biolector automated fermentation platform. Because of the high quality that it produces, we then assayed production of flavulin, which in this case can be optically measured. We put that data into the experiment data depot, and then ART drew that data and made some predictions on how concentration relates to flavulin, and made concentrations. Well, it made recommendations on the concentrations for the next DVTL cycle. Those recommendations were translated into robot movements through a Jupyter notebook, and then started the next synthesis media, and that continued for 70 DVTL cycles. You go to the next slide, you'll see that the first time we tried it, we did a couple of DVTL cycles to train ART, and we got an R squared, a coefficient of determination of around 0.32, which is not fantastic. But when we asked for recommendations from ART, and next, we saw that those recommendations increased the production of flavulin titer by around 148%. And if you go to the next slide, you'll see that we've done this several times. So you go next. This is the top one is the one when increased 148, and the next one it will be another set of DVTL cycles. We're trying to increase titer, and we were able to increase it by 170%. And in the next one, we used it to increase yield, and it increased yield by around 300%. If you go to the next slide, you will see that surprisingly, the media component that was most important to explain production and to increase it was sodium chloride. And it actually, the algorithm, the active learning process that we used with ART took us to the maximum that Pseudomonas could actually support, around 400 millimolar, which is close to what you get in seawater. So that gives you an idea of some unexpected insights that you can get by using machine learning in well-known systems such as Pseudomonas putida in this case. Next one. As I said, ART can take a lot of different inputs. And we're using a variety of projects. So for example, one of them is choosing CRISPR-I targets to improve the titer of sustainable aviation fuel, isoprene. Next. We're also using it to make the first quantitative predictions of polyketide synthesis. We take as inputs there the features from the sequence and the phylogeny and chemical structure of polyketide synthesis, and we're able to predict for the first time, I think, quantitatively what is the production of those polyketide syntheses. Another one, next one, it's being used in a microfluidic self-driving lab, and we use it to optimize, to find the optimal field, voltage, and reagent concentration to maximize transformation efficiency. And we've been able to increase the amount of colony-forming units by around twofold. Next one. We're also using to predict scale-up, and we use biolector data and biolector production data to predict Amber 250 production data, showing that we can use some of these tools to predict how cells bioengineered cells can perform at higher scales. So in general, what you see is that ART is a generally applicable tool to many synthetic biology and biomanufacturing problems. If you want more detail, there is an ART website with tutorials, and next. And also publications next. And among the many projects that it has been used next, it's an iGEM competition in which the Dutch team used it to engineer a Saccharomyces species to produce alpha amylase. If you want more details, you can go to art.lbl.gov and you can see the Nature Communications publications and some of the other projects that we're developing and in which we're using ART and some of the upcoming publications. So thank you very much.